Okay, starting the recording. All right. So where we left off before the break, we were going over outputs from RSEM, which is the tool that we use to quantitate our RNA-seq data at GeneLab. So as I mentioned, RSEM will output two quantitation files, one to estimate expression values per gene. That file ends in genes.results, and one to estimate expression values per isoform. That one ends in isoform.results. For DGE analysis, differential gene expression analysis, we're going to be using the estimates per gene. So this is an example of what that file looks like. And so similar to what I've done with other files that look like this, we'll go through this column by column to let you guys know what information is provided in the genes.results file. So in this first column, you'll see the gene ID, which is the gene name according to the database used. And of course, since we used Ensemble, we see Ensemble IDs here for the mouse, and that G here that you see in the ID is per gene. The second column is going to contain the transcripts IDs for that respective gene. And so you'll notice here, again, this is going to be from the database that you used for your reference, which for us was Ensemble. And so this is the transcript IDs. Have a T here instead of a G. It's how it distinguishes between the G and the T. And of course, genes can have multiple isoforms, which would be multiple different transcript IDs. And if there is multiple isoforms, you'll see each one of the transcripts that are derived from that gene separated by a comma, as you see in some of these genes here. Oh, I should also indicate that the line with the star, so gene ID that ends in 56, the values for each one of these columns I have in the brackets here. So this gene ID here again ends in 56 when we're looking at. Then you'll notice that this particular gene has 1, 2, 3 transcripts associated with it, or 3 splice isoforms of the gene. And then the third column is going to be the length of the gene. So this is going to be a weighted average of the respective gene's transcripts length, right? Because each one of these transcripts, depending on the length of the exons that are included in that transcript, will be a different length. And so this initial length column is looking at the weighted average, assuming that all of the transcripts are expressed equally. And then the fourth column, the fourth column is going to be the effective length. So I should say for this example one, the length is 3,894.46. And then the effective length, you see is slightly different than that. So the effective length is going to be the weighted average of the respective gene's transcripts effective lengths, which are weighted by each transcript's isoform percentage. So this will actually take the information about the expression of that transcript from your data, and depending on the level of expression of each of the transcripts for a particular gene, it'll modify the length of the gene relative to the percent expressed for each isoform, because of course those isoforms are going to have different lengths. Our fifth column here is the expected count column. So this is the sum of the number of read fragments. So for single end data, it would just be the one read that we have. For paired end data, this is going to be the full template. They're calling it a fragment here, that were derived from each transcript of the respective gene. And this is the value that we're going to be using as the raw expression value when we go into our differential gene expression. And so for that highlighted gene 56 here, we see that raw expression value is 2,108. So that's the number of templates that mapped to that gene. And then it also gives you the quantitation for each gene in some normalized manner. So column 6 is in transcripts per million. So it's going to be the relative measure of transcript abundance. And this value is going to be summed over all transcripts for each respective gene to generate the gene transcripts per million. And we're going to get into that a little more later in the lecture. And then Lauren's going to go over different normalization methods in the statistic lecture as well. And then the last column that we get in this genes.results file is the expression, the raw expression, this time normalized in fragments per kilobase of transcript per million mapped reads. So it's essentially another relative measure of transcript abundance. And again, this value is going to be summed over all transcripts for each respective gene to generate that FPKN value. And just for that gene of interest that I started here, gene 56, we see that the expression in TPM is going to be 3.57, expression in FPKN is 8.86. Questions about any of these columns? We're going to be looking at them during the Jupyter Notebook portion.
Yeah, I have a couple of questions. Why is it called the expected count instead of the estimated count?
I don't know. Does anyone know why the developers named that column expected count?
Okay, and then I'm a little confused because of the effective length is the average.
Mm-hmm.
And does it select the longest transcript and says that's the length? Because it says this is the length, and then it shows an effective length, which is the weighted average of the isoform lengths.
Mm-hmm.
So what's in the first column for length?
So the first column for length is going to be the average length if each of the isoforms or each of the transcripts for that gene are all expressed in equal proportions.
Okay, okay. And then the other is weighted based on
Based
distributing them.
the actual data. So the effective length is looking at the actual data. So the effective length basically saying if we look at the second gene here, we have it only has two known isoforms, two annotated isoforms here. So this transcript ending in 3 and ending in 41 here. So in this case, the length would essentially say that this transcript and this transcript are equally expressed. Of course, that's usually not the case for isoforms. We usually have one or more isoforms expressed more than the others. And so the effective length actually looks at the data to see how much over all of your samples, how often is this transcript expressed, how often is this transcript expressed, and then uses the actual percent expression over all samples to weight the length. So, if one of these is a little bit longer than the other, and that longer one is expressed more, it's going to be a little bit longer. If the shorter one is expressed more, it's going to be a little bit shorter, and looking at the averages.
It's so interesting in these examples, the effective length is always shorter, which seems there's a bias towards shorter versions of the isoform, but I
Yeah,
guess it's just a coincidence. Thank you.
for sure. Any other questions about this? We're going to be looking at and interpreting these from our actual data. Okay. And so once we have those data from all of our samples, we need to put it all together in a matrix or in a table so that way we can start analyzing them all together. So after we have these files for each of our samples, we're essentially going to grab the expected count or the number of templates that mapped to each location, to each gene, and we're going to put it in a table such that all the genes are listed in the first row of the table, and each sample is going to be, or each column is going to be a different sample from our study. So for us, we have 6 samples from flight, 6 samples from ground control. I don't know why I'm only showing 5 and 5 here. I should update that. And you see just the gene that we had been following, the one that ends in 56, we had that expected count value is 2,108. So when we put it in a table, we would see that for rep 1, that's 2,108 there in the table. Any questions about how we quantitate data before we do it?
Amanda, I have a question.
Yeah.
So all this data recorded are counting both sense and antisense. Ideally, it sounds like you should count only the sense, but our data have majority in the antisense. So how can that be happening, and how do they solve this problem?
Great question, Maijan. So in the RSEM calculate expression, one of the options that you indicate is strandedness. And this is where you'll tell RSEM if your data are stranded, and if so, in which direction. So if your data are not stranded, you would set this value to none, and that's actually the default. And in that case, they would count templates align to both the sense and the antisense of your reference. If you looked at your data and it was all, the majority of it was in the same direction or the sense orientation relative to your reference, you would use a forward setting for this option, and that would tell RSEM only count templates that are mapping to the same direction as the reference. But for our case, we know that our data are antisense relative to the reference, so we'll set this option to reverse, and that's going to tell RSEM to only count the templates that are mapping to the to antisense relative to the reference.
Okay, that makes sense. But why is our data in antisense format? Is that because of the library prep?
Yes, it's because of the library prep and how the strandedness is achieved for that. And I will tonight work on that slide that Elva mentioned to try to follow this all the way through from start to end to make that more clear. But yeah, it's due to how the library prep allows the samples to be stranded.
Okay, that makes sense. Thanks.
Of course. Any other questions about how we quantitate with RSEM? Okay, the questions come up. Of course, feel free to unmute your mic and ask, put it in the chat. In the meantime, we will hop back to our Jupyter notebooks. So everyone go ahead and bring back up your Jupyter notebooks. Make sure that you see in the bottom left that you are running a Python kernel, conda environment :GL4U, and that it's idle. If anyone's seeing anything other than this, please let me know. And so where we left off here is we finished assessing the strandedness of our data. This is how we know that our data are stranded and that the templates are in the antisense orientation relative to the reference, which, as Weijen just noted, is pointed out in the RSEM algorithm. We actually have to tell RSEM what strandedness our data are. So as I mentioned in the lecture, before we can quantitate with RSEM, similar to STAR, requires an index of the reference genome that we're working with. And so we're going to generate the RSEM index using the RSEM prepare reference program, and that's going to be from the GTF annotation files that we get from Ensemble, as well as the associated FASTA files. Again, in GeneLab, we use the Ensemble database for that. And to save time and storage space, this only has to be done once, so I did it for us when I was going through and processing these data. But just to go over it, this is, again, the command that was used. So we use the RSEM prepare reference, which is how that is called in the RSEM program. We're going to give it this --GTF option, which is going to indicate the location of our reference GTF file, our gene annotation file for mouse. And then it expects two positional arguments, one before the last being the name of the FASTA file, which contains the genomic sequence of our reference. And then in the last position of the command should be the location of where we want to output the RSEM index. And then if you want to put a prefix to those output files, you can. At GeneLab, we always do the first letter of the genus and then the first three letters of the species name for our prefix for the RSEM index. And then, of course, those definitions are all in the RSEM documentation, which is linked out to here. You don't have to know them. And for all the ones that we use in this notebook, I did indicate all of the definitions for each of them right in the notebook, so you don't have to hop back and forth to the documentation. The input data to prepare the reference, is, again, similar to STAR, is going to be the FASTA file containing the genomic sequence. For us, that would be the mouse genomic sequence, and then the associated GTF, or annotation file in gene transfer format. And then RSEM will output several different files here, and these all start with MMUS because that's a prefix that we told it. And again, this is just organizing that annotation information into certain slots within the index, so that way when RSEM does the quantitation, it can easily find those annotated regions. So again, only has to be done once, and then we're going to need it to be called when we actually do the quantitation with RSEM. So moving on to step 8A, which is counting the aligned reads with RSEM. And so I know we kind of went over this, but just to refresh, similar to read alignment, quantifying the number of reads aligned to each gene can also be pretty challenging. Why is that? You guys can put in the chat. I know the answer's right there, but put it in the chat, unmute your mic. Why is it challenging to do this quantitation? No one wants to answer? The answer's right there in the notebook. Why is it challenging to quantitate our aligned reads? No one wants to tell me? Okay.
Because of the significant sequence overlap?
Yeah, because there are reads that can be mapped to multiple locations on your reference genome. And if it does map to multiple locations, how are you supposed to know where it comes from? So that's a big issue when it comes to quantitating. And similar to genes, there's also can be different splice isoforms. If you want accurate isoform quantitation, you also have to deal with reads that could map to multiple splice isoforms of a gene. And so as we went over in the lecture, the RSEM software package was designed to address these issues and rescue those reads that are not uniquely mapped by allocating them or a portion of them to the transcripts that they're mapped to using the 3-step algorithm. So again, the first step is that RSEM only looks at reads that are uniquely mapped, so only looks at reads that come from one gene, that map uniquely to one gene and one isoform of that gene. And then for each reads that map to multiple location, and then we'll do maximum for each read map to multiple location, and it will divide it between the transcripts to which it maps, proportional to their abundance estimates in the first step. And so after it looks at the uniquely mapped reads, it'll estimate the abundance of the transcripts based on those uniquely mapped reads, and then it'll go through and look at the multi-mapped reads, and based on the abundance estimates of the transcripts in the first step, it'll allocate a portion of that read to one or more of the genes that it maps to. And then it'll compute the final abundance based on the updated counts for each transcripts, now incorporating in those multi-mapped reads. So that's why, again, after a lot of debate with our AWGs, we settled on this one because it does recover those multi-mapped reads so they're not thrown away, and how they are allocated is based on the data and what the expression of the uniquely mapped transcripts or uniquely mapped reads look like. So we're next going to estimate gene and isoform expression levels for all 12 of our samples using the RSEM command that I went over with in the lecture. And again, you can review any of the options in this command using the RSEM user guide for the calculate expression algorithm of RSEM. So links out to there. And of course, this takes a while too, not quite as long as STAR, but still takes a little bit. And so we will be submitting this to a Slurm job scheduler in an array because, again, all 12 samples will have the same command run. And so this is all of the options that I went over with in the lecture. I am happy to go over any of these. Again, the only one I didn't go over is the number of threads. That just tells the system how much compute resources are available for it to use for this. And all of those parameter definitions are, again, given in the Jupyter Notebook here. So you'll see those definitions again, both in the Jupyter Notebook as well as in the lecture. And then as Wygen pointed out, we have to tell RSEM the strandedness of our data using that strandedness option. And we know from performing the RSeq QC infer experiment in step 6 that our data are anti-sense relative to the reference, and so that setting for RSEM is going to be reverse. So it'll quantitate only the templates that are mapping in the anti-sense direction relative to the reference. And for RSEM, for the RSEM calculate expression, this is called for the RSEM calculate expression command, it requires 2 different input data. So it requires your RSEM genome reference or index, which was made in the previous step. And then it also requires, we're quantitating transcripts. So this is going to require the align to transcriptome BAM output file from the STAR step. And our output data that we're going to get from RSEM is we're going to get a genes.results file, which we went over in the lecture. We're also going to get an isoforms.results file, which we did not go over in the lecture, but we'll take a look at here. And then we're also going to get a directory with 3 different files in it, which indicates statistics of how the RSEM quantitation went. And so with that, I want everyone to submit their Slurm job to the simulated job scheduler by running this xbatch command here. And so again, this is just setting up an array script such that each of our samples will run through the same exact command. And so while those are all mimicking being run, we're going to take a look at an RSEM standard out file. This is a pretty large file, so we can go ahead and take a look at what the standard output of RSEM looks like. And so again, a lot of this stuff up front, I should ask, is there anyone who's not seeing the same output that I'm seeing here? Okay. So, of course, I always add that extra stuff to my Slurm scripts, which you're seeing here, to just print out the name of the job that I'm running, the start time of the job, the server that it was run on, the array task ID so I know which job number is being run. I also print out the sample name so I can keep track of which samples are being run. And I also output the version of the tool that I'm using. And then I also echo out the exact command here. So this command, just to make sure that it was indeed filling in that sample variable with the sample that it's working on. So in this case, our flight rep1 sample. And then starting here is the standard output of RSEM. So you'll notice that it gives you a warning about the fact that we're using read lengths of less than 25. So again, 25 is the default for RSEM. So it just gives you a heads up that, hey, we don't recommend this, which is fine. And it actually doesn't allow it if you're using poly-A data. So if you used mRNA enriched data, we'd have to change this. But we're not. We're using our deplete. So 20 is acceptable, even though it gives you a warning. And then it basically just goes through and gives you a log of the output. So how many entries or templates that it's looked at so far. And so that's why this is very long. And then this is more information going through, now it's going through the index and the different annotations within the index. So looking at different aspects of the genes that are identified in the index, and it'll go through and do that and give you log output. So I kind of like skim through these. It's basically just going through and saying that it's quantitating the different gene annotations that it has in the index. And so I kind of just skim through this just to make sure I don't see any errors or anything like that. This is doing the estimation, so the maximum likelihood estimations, and it's telling you how it's running those algorithms. Again, looking at the uniquely mapped reads and creating abundance estimates based just on the uniquely mapped reads first, so it goes through and gives you a log status of all that. Again, a lot of information. I pretty much skim through it to make sure that I don't see any errors or warnings. But yeah, it's pretty long because it's going through and doing this estimation for all of the genes. And we know that in the mouse, there's what, 20, 30,000 annotated genes now. So you can kind of skim through all of this to the end, and then we'll collapse it when we get to the end. And so basically at the end, after it goes through and does all of the quantitations, it'll tell you that your expression results are written. This one also gives you a timestamp for how long it took. So this one was 31 minutes. And then we also see here this rm-rf. So it creates a temporary file in order to do the abundance estimates and maintain it to be able to call those abundance estimates when it's determining where to put those multi-mapped reads, how to quantitate those. So essentially all those temporary files, the program will remove after it finishes. So yeah, pretty much you're just looking to make sure that the expression results were written, it finished successfully. You don't see any errors or any errors within the output. And so obviously that is huge. It takes up a lot of space. So if you guys want to, I'm going to do this. Click on this blue here, and it will collapse the output of that RSEM standard out. And let's take a look at some of the output files that are generated, and then we're going to answer some questions. So we're going to take a look again at just the first few lines of the genes.results file. So because we're just looking at the first few lines in the genes.results file, it's not compressed, it's a standard text file, so you don't have to do anything fancy to open it to view it. You can just use your head command. How does the number of threads affect the time and space? Yeah, so the more threads you have, so basically the number of threads is how it can split up the data and the information that it's trying to generate. And so the more threads that you have, the more it can split the data up and run things in parallel. And so the more threads that you have, the faster it'll run. So again, the threads are just dependent on the compute resources that you have available. So you want to give it the maximum number of threads that you can per sample. So here, we're just going to take a look at the first 10 lines because we're running the head command, and the exclamation point means we're running this in Unix of our genes.results file. Again, we're just looking at one of the samples, so the flight rep 1. So you guys can go ahead and run that. And you should see 10 lines in total from this output. Yeah, cool. So I'm just going to shrink this down a little bit. Try to get these views. So again, this is the file that I went over in the lecture. So we have our gene ID, we have our transcript IDs, the length, the effective length, the expected count, and then the count calculated as transcripts per million and calculated fragments per kilobase per million. And just as a reminder for what each of the columns mean, I have them listed here. So listed here, the definition of what information can be found in each one of these columns. Same thing I went over in the lecture. So again, I have some redundancy here, both in the lecture and the Jupyter Notebook, we see those definitions. And so I want you guys to take a look at this output file from our data, and I want you to answer some of these questions. So the first question, Armin, if you could cue it up, is what is the first gene listed? And Armin did put the, yeah, thanks. So Armin put the link in the chat again. So what's the name of the first gene listed? Just go over, sorry, I can see your answer, but the first gene listed would be indicated in the gene ID column, not to be confused with the transcript ID. So the first gene listed is going to be in the gene ID, or the first column of this file. And we see here that the first gene listed is going to be the ensemble mouse gene that ends in O1. So for those guys who had that answer, that would be the correct answer. Let me know if it's unclear how I got that information. And then, oh, you looked it up, Alba. We're just going with
How could I not? It's number 1.
We're gonna add annotations in the DGE portion with Lauren. So we will convert all of these ensemble IDs to gene IDs and symbols that make more sense. So go ahead and answer the question that Armin should have up now. So for that first gene, what is the gene length? Yeah, for that first gene, what is the gene length? Okay, hurry up and submit your answer. I'm going to go ahead and show the results. So for that first gene listed, what is the gene length? So yeah, for those of you guys who put 3,262, that is correct. Someone thinks that it's currency. I don't think so.
Good one.
Yeah, so some of you guys put the actual gene name, maybe you're answering the previous question. Yeah, so that first gene, what is the gene length? So the gene length is in the third column of the output for this genes.results. And so gene ID is going to be in the first column, the transcript IDs, and these are tab separated, so you'll see spaces in between them. The transcript ID is the second column, so that third column is what we're looking for the first gene listed to find the length of the gene. So that would be 3,262. Our next question is again for that first gene. What is the expected count value? So Aaron, if you could put that question up, and then if you guys want to answer in that Menti survey for what is the expected count for that first gene listed. Okay.
All right, we have 8 answers.
Okay, let's take a look. A lot of 869, 3062. Okay. Okay, so what is the expected count? So again, taking a look at the columns here, the expected count value is going to be the 5th column. So we're looking for the value that's in the 5th column for that first gene listed. So again, the first gene listed is going to be here. The first column is going to be the gene ID. The second column is our transcript IDs. Third column is the length. The fourth column is the effective length. So I think someone accidentally got the value for the effective length. And then it's the 5th column that's going to be the expected count. So for this gene, the expected count is 869. For those of you who didn't answer or or got this answer wrong, is it clear how this information was found? If not, let me know. Okay, move on to the next question, which is what is the transcript per million value for that first gene? So looking at the expression in transcript per million for that first gene. So go ahead and submit your answers in the Menti. Look for one more answer, hurry up and submit it. It's okay to put I don't know if you don't know.
All right, voting's closed.
Okay, let's take a look at these results. So a lot of 5.95, which is correct. I agree that NASA is the best. So yeah, you guys all got this one right. So again, just looking at the definition of these columns, we know that the expression value is going to be found in the expression value in TPM or transcripts per million is going to be found in that 6th column. And so looking at this first gene, again, gene ID, transcript IDs, length, effective length, expected count in the 5th column, and then that 6th column is going to be the expression in transcripts per million. So 5.95 was indeed the right answer. It looked like at least everyone who answered got that one correct. And so now question 3, I want all 3 of these values that you just gave me for the first gene. I want you to give me those same values for the 3rd gene listed. So the 3rd gene listed is going to be 1, 2, 3. It's going to be this gene ending in 28 here. And so for that gene, 3rd one down in the file, I want you to give me the gene length, the expected count, and the expression in transcripts per million. So looking for 3 responses for this question. Okay, in the interest of time, I'm going to go ahead and show the responses. So, let's see. Some people put gene length, expected count 91, TPM 1.5. It's looking like a lot of you guys put that same response, except for one person who might just be reading the wrong line. Yeah, most of you guys are correct. So again, looking at that 3rd gene down, we want to know the gene length, the expected count, and the expression in TPM. So the 3rd gene listed here ends in 28. So again, going through the columns, first column gene ID, second column is going to be a comma-separated list of all of the transcripts that can be derived from that gene. Third column is going to be the length. So a lot of you guys got this right. So the length for this gene 28 here is going to be 1,863.71. And then the next value is the effective length. And then the value after that is what's going to be, and it's split on my screen, sorry about that. So the next value here is going to be the expected count, which is 91. And then we have the expression value in TPM, which is 1.15. Is there anyone who's still unclear how we arrived at those values? Okay. Let's go ahead now. And I know I didn't go over this, but I do want you guys to take a look at this other output from RSEM. So in addition to taking a look, or in addition to quantifying this in the context of genes, RSEM will also spit out a quantitation table for the isoforms. So if we go ahead and take a look at the first few lines of the isoforms.results file for that same sample we've been looking at, flight rep 1. I want you to take a look at this, and I want you to answer some questions here. So the first question is to calculate the sum of the expected count values for all isoforms associated with the gene that ends in 28. So that's this gene right here. And so you see, notice now that in the isoforms.results file, we have the transcript ID first and the gene ID second. So if you recall from the genes.results file for this gene here that ends in 28, there are 1, 2, 3, 4 transcripts. So there's 4 splice isoforms for this gene that ends in 28. And you'll notice in the isoforms.results file, we have the transcript ID listed first and the gene ID listed second. And so you want to calculate the sum of the expected count values for all isoforms associated with gene 28. So as we saw in the genes.results file for gene 28, there are 4 isoforms. And so we should see gene 28 listed 4 times in the gene ID column since we're going based on their transcripts ID. So again, 4 isoforms or 4 transcripts from gene 28. I want you to sum up the expected count values. So looking at the columns, we have transcript ID is the first one, gene ID is the second one, length is the third, effective length is the fourth, and the expected count value is the 5th column. So we're looking at that 5th column, again, summing the values for all isoforms associated with gene 28. So go ahead and put your calculations. You could just put the sum. You could put what the calculation would be. I'll take either of them into the link that Armin sent. Awesome. Okay, let's take a look at what you guys put. 91, 298, 76, 91, 74, and 69 times 4. Interesting. Okay, so 91 is the right answer. I see that there's a mix, about only half of you got that. So what we want to do is, unlike the genes.results files, which is focused on the gene ID, so the gene ID is the first column here, and then the transcripts IDs are the second column, and they're listed in comma-separated manner. For the isoforms.results file, we have the transcript IDs listed first, and the gene IDs listed in the second column. So that means that for gene IDs with multiple transcripts, we're going to see that gene ID listed multiple times, once for each transcript, right? So transcript ID is making up our rows here. And so for gene 28, just like in the genes.results file, we saw for gene 28, we see 1, 2, 3, 4 transcripts listed. And so we see here, for gene 28, we should also see 4 transcripts listed. And indeed, if you take a look at these columns, we see gene 28 listed 4 times, once time associated with each of those 4 transcripts. And so we wanted to sum the expected count values for all of those isoforms or transcripts listed here. And so you would go to the expected count column, which we know is the 1, 2, 3, 4, 5th column for each of these genes. So this column here is going to be the length, this column here is going to be the effective length, so this column here is going to be the expected count. So I was looking for 74.69 from this first transcript of gene 28 plus 10.20, which is the expression for the second transcript of this gene 28, plus 6.11, which is the expected count of this third transcript for gene 28, and then plus 0. So this fourth transcript for gene 28 is obviously not expressed in the sample, so we have a 0 count there. So if you sum up 74.69 plus 10.20 plus 6.11 plus 0.00, you will get 91.00. And so that is how we end up with the answer of 91.00 for the expected count for all transcripts associated with gene 28. Is there anyone who is still not getting how we got that value? Okay, great. If it's still not clear, let me know. And then last question in this section is now I want you to tell me how this value that we just calculated compares with the expected count for that gene 28 in the respective genes.results file that we looked at up above. So how does that value, the sum of the expected count values for each of the 4 isoforms for gene 28, how does that sum of all 4 of those isoforms compare with the expected count that's listed for gene 28 in the genes.results file for the sample? So go ahead, Armin, if you want to give them a minute to respond.
Voting's closed. We have 7 answers.
Okay. They're the same, they're the same, they're the same, they're the same. Yes. Someone was away from their computer, no problem. Or keyboard. Yeah, they're the same. So that makes sense, right? So the sum of the expected count values for all isoforms associated with this gene, as we just calculated, 74.69 plus 10.20 plus 6.11 plus 00 gives us the value of 91.00. So since that is the sum of all of the transcripts that were expressed in this sample for this gene, then the sum of the expression of all of the transcripts for the gene should match the total count of that gene. And sure enough, it does. So if we take a look at gene 28 here, the expected count value is going to be in the 1, 2, 3, 4, 5th column. And so looking at the 5th column, the 3rd, 4th, 5th column, it is 91.00. So the expected count of a gene is going to be the sum of the expected counts for all of the isoforms of that gene. For those of you guys who didn't answer, or for that person who is away from their keyboard, do you need me to go over this again or in a different way? Is everything clear? If it's not clear, let me know. Awesome. Okay, now that we know how to interpret these genes.results files and also the isoforms.results files, so we do provide both of these outputs in GeneLab data. Why are some counts in decimal? Great question, YGen. So when we did our quantitation for arsine, we do our quantitation in a 3-step process, right? So we first estimate the abundance based on uniquely mapped reads only. Of course, a uniquely mapped read, one template, if it maps to one location on the transcript, that template, wherever it maps to on that gene uniquely, is going to be counted as 1, right? One template mapping to this location, right? If we have 5 templates mapped to the same gene in a unique way, then there will be 5, uniquely count of 5 for that gene. But then after it estimates the abundance based on uniquely mapped genes, it then computes the maximum likelihood estimation so that for genes that are mapped to multiple locations, so in the example here, for genes that are mapped to multiple locations, like this purple gene in the example I showed in the lecture, it'll use the likelihood based on the expression of according to the uniquely mapped genes depicted here in black. Then it'll calculate where this template should be assigned to, and if it should be assigned to more than one of the genes that it maps to based on those uniquely mapped reads, it'll split this up as a fraction. So in this example here, looking at the uniquely mapped templates to gene A, we see we have what, 8 uniquely mapped template. So that would be an expected count of 8 to gene A from just uniquely mapped reads. And uniquely mapped read to gene B here, the expected count would be 1 in this example. So then it goes back in that second step and it calculates, or the third step I should say, is it calculates the abundance of these uniquely mapped reads based on, or of this multi-mapped read, excuse me, based on the uniquely mapped reads. So in this case now, when it goes back and tries to allocate this multi-mapped read in purple here to either gene A or gene B, well, based on the maximum likelihood estimation of these uniquely mapped reads, it seems like it's more likely to have come from gene A because gene A is more expressed based on the uniquely mapped reads than gene B. And so what it does now is it splits up this read into a fraction, and it'll put the majority of that fraction, I don't know if it's true, but let's say 0.8 of that read will go and be associated with gene A, and it'll give 0.2 of that read associated with gene B. So then the final expected count of gene A in this example would be 8.8, and the final gene count for gene B would be 1.2. So that's why we're getting those decimals, because of those multi-mapped reads. Does that make sense, Jen? Okay, great. Yeah, so if we would use one of those quantitation methods that doesn't do a weighted quantitation and just throws away all of the multi-mapped reads or just counts all the multi-mapped reads as one, then you wouldn't end up with any decimals. But because we use RSEM with the maximum likelihood estimation, you will see some of those raw count values or those expected count values with decimals because of that. Any other questions about RSEM output files or the expression information provided in them? Okay, we will move on to 8B, which is to compile our count data with multi-QC. Again, we just want to make sure that all of our count data looks good, looks as expected. We're able to quantitate each of our samples. And so we're going to take a look at one of the RSEM output files that essentially summarizes the quantitation results. And that's the RSEM CNT stats file, which is outputted in a directory that's made from RSEM called stat. And in there, remember I mentioned there are these 3 files. So this CNT file contains a summary of how the quantitation went. So kind of similar to that log final out file for the star alignment that summarizes how the mapping went. This is going to summarize how the quantitation went for each sample. And so we're going to go ahead and just right off the bat compile the information in that CNT file for all 12 samples, again, using multi-QC. So RSEM is another set of outputs that multi-QC can recognize. And so I know we're all familiar with this command now, so start with multi-QC. This time we're going to give it a prefix with this -n option called count, because this is the multi-QC for the count data. Output directory in that subdirectory that we made right at the beginning under the RSEM counts folder. And then this is going to be the path to where that CNT RSEM output file exists for all samples. And so if you haven't done so already, go ahead and run that command. Okay, is there anyone who's still waiting for theirs to finish? Is there anyone who doesn't look like what I'm showing on my screen here? Okay, so we're all used to this multi-QC standard output by now. What we're looking for in this case is that RSEM files have been identified. And of course, we have 12 samples, so we assume that they would find 12 RSEM reports, which they do. And we don't see any errors popping up, and we do see that multi-QC is complete. So that gives me confidence that we can view the multi-QC report. Again, parameter definitions listed out here. Input data in this case is that CNT file, which is going to be a summary of the alignment info and stats that were used for quantitation. And then the output data, like we're used to with multi-QC, a folder containing the information, that CNT file for each sample, and then those data plotted in plots in an HTML report. So let's take a look at that HTML report. So we'll go ahead and load our Python library needed to show it, and then we'll go ahead and run this iframe command to view the multi-QC report of our quantitation statistics. So this is information that's grabbed from those BAM files based on how it was quantitated with RSEM. So what we see here is the percent of alignable data that was able to be quantitated. And again, similar to the other multi-QC outputs, we can toggle to sort this for highest percent alignable versus lowest. I should say here, this is a really good check if you put the wrong option for the strandedness for RSEM. So if it only quantitates the templates that were aligned in the wrong orientation according to the strandedness, this percent alignable value will be very low. And so that's how you know that you made a mistake, likely in your strandedness setting. You need to go reassess that. We also see here, it breaks it down based, again, on the information that it found in the BAM file. This is raw numbers, or we could do percentages if we want to more easily compare each sample. So samples here on the left, percentage here on the x-axis, and then color-coded. So in blue here is going to be ones that are aligned uniquely to a gene, so templates that were aligned uniquely to a gene. In light pink is going to be templates that were aligned to multiple genes. In red here is going to be templates that failed to be quantitated because it aligned to too many locations. And then we don't really see it in here, but, oh no, this is unalignable reads. So failed due to too many alignments is the color that we're not really seeing here. And then this dark maroon is going to be the number or percent, in this case, of reads that were unable to be aligned, mostly because they were not mapped to any place on the reference. So I want you guys to take a look at these values and answer. Oh, sorry, I almost forgot. There's also mapping rates here. So this is going to be a histogram showing how many reads were aligned to reference regions. And so this is another way of looking at the total number of alignments based on the count. So you see the majority of them aligned to one or two places, and you can hover over to see that information for each read. So it'll tell you how many just had one alignments, and then you have some reads we're going to align to multiple locations. So how many alignments it is how many places that it aligned to on the reference that were quantitated. That's the last thing on the RSEM multi-QC. So I want you to take a look at those files, and I'll give you a minute for each one of these before we head into our lunch break. So Armin, if you want to start that first question here, which is which sample has the highest percent alignable reads and which sample has the lowest? So the highest percent of alignable reads that were quantitated and which sample has the lowest percent alignable reads that were quantitated. Okay, so we're just going to go over these answers in the interest of time. I know we're already at our lunch break. So basically for this, which sample has the highest percent alignable reads and which sample the lowest? If you scroll up to the top of this graph, or of this HTML report, we'll see here the percent alignable reads is nicely indicated for each sample here. And so you again can click on it to sort. And so if I sort from highest percent to lowest, I'll see the sample with the highest percent alignable reads is our rep 2 of our ground control group. And then sorting in the reverse direction, so the least percent alignable reads, which is 87%, which is still really good, is going to be our ground control rep 6. And then for that next question, which sample has the highest percent of unalignable reads and which sample has the lowest, that's going to be the inverse of what we just said for percent alignable. So it'll be ground control rep 6 has the most unalignable reads at 13%, right? 100 minus 87. And then ground control rep 2 will have the least percent of unalignable reads. So again, just 100 minus 90.5 would be 9.5% of unalignable reads. That said, we're about a minute into your lunch, so go ahead and take your lunch. When we get back, we'll quickly go over that one more time, and then we will generate our raw counts table, and then we're going to spend the rest of the time doing a debrief on this first Jupyter notebook. So enjoy your lunch, and please be back here by noon Pacific time.